Hello and welcome to this video on multiple alleles and the ABO blood group in human. By the end of this lecture, you should be able to understand the concepts of gene locus, allele and multiple alleles. In addition, I will explain the concept of multiple alleles of the ABO blood group system in humans. So let us discuss genes. Genes are same as Mendel's factors. Mendel did not use the term genes, alleles, chromosome, DNA because this terminology was not invented. It came much later after he published his result or findings. Genes are located on chromosomes. For example, a human cell contains 23 pairs of chromosomes which are shown here. This is known as karyotyping, in which chromosomes are arranged according to their size and bending pattern. So there are 23 pairs of chromosome. Out of the 23, 22 pairs are autosomes. Most of the phenotype and diseases are caused due to genes located on autosomes. Therefore, most diseases are autosomal, or most phenotype are autosomal in nature. The 23rd pair is XX in female and XY in male. So this is a male karyotype. As you can see that XY pair, Y is very small, it has less than 300 genes. Therefore, you will appreciate to know that mother donates more DNA to male child as compared to father. Each pair is a homologous chromosome because one set is coming from paternal side, the other set is from maternal side. So such chromosome pair is known as homologous chromosome. So now genes, how the gene is defined. Gene is a functional part of chromosome or DNA that encodes RNA, protein or phenotype. I have shown here one pair of homologous chromosome where four genes are shown, A, B, C, D. So whatever genes you have from paternal side, same genes are inherited from maternal side as well. How many genes are there in human genome? There are 25 to 30,000 genes total in human genome, which together makes about less than 2% of human genome, which is called as euchromatin. The remaining 98% of human genome is non-functional and called heterochromatin. Gene locus. Position of a gene on a chromosome is known as gene locus, or plural is loci. Every gene has a fixed locus or fixed position on the chromosome which does not change. However, some gene can move from one place to another. Such genes are known as mobile genetic element or mobile genes or transposons, and this process of movement is known as transposition, which is rarest and causes abrupt phenotypic changes and diseases like cancers. I would like to explain allele, which is a difficult concept and may be confusing, so I would like to draw your attention. Here I have shown a homologous chromosome pair showing maternal and paternal chromosomes. So there are four genes A, B, C, D which are shown here. Same genes are also present from in the maternal chromosome. So basically at each locus there is an alternative form or a variant of the gene. This is known as allele. So an allele is an alternative form or a variant of a gene located at the same locus on the homologous chromosome. Two forms of a gene at a locus is known as allele pair. So these are four pairs of allele I have shown here. When allele pair in a homologous chromosome on a given locus is identical, dominant-dominant or recessive-recessive, that condition is known as homozygous. On the other hand, when the allelic form of a gene at a given locus is different, one is dominant, other is recessive, that condition is known as heterozygous or hybrid. So now I would like to go over multiple allele. Mendel in his experiments used contrasting phenotypes for each trait. For example, for height he selected tall or dominant, dwarf or recessive. However, if you go to nature, you will find that the height of pea plant changes gradually, and each having its own unique allele pair. So later scientists discovered that in nature more than two allelic forms exist of a gene. So each plant has a unique allelic form which is different than the other one. Presence of more than two allelic forms of a gene in a population was called as multiple alleles. In fact, every gene now has multiple forms. That is why we look different from one another. However, an individual animal can have only two allelic forms of a gene. In the next slide, I will explain further. I will elaborate multiple alleles further with an example of ABO blood group. I would like to discuss multiple allele further with an example of ABO blood group. ABO is determined by the presence of glycoprotein antigens A and or B on the RBC cell surface. ABO is encoded by an I gene. So there is one gene, however it exists in three allelic form, IA, IB and IO. Therefore this is an example of multiple alleles. When A is expressing on the RBC surface, it gives A blood group type. When B is expressing on the surface, it gives B blood group. When A and B are expressing, it will give AB blood group. When none is expressing, it will give you O blood group type. However, an individual can inherit a pair of these blood group alleles. So the question is, how many ABO genotype or alleles exist in human population? So how many ABO blood group genotypes exist in human population? We will use Punnett square method. So you have allele ABO from female and these are from male. So 3 by 3, 9 is small squares. Put the combination and predict the phenotype. Now, so there are total 9 ABO genotypes in human population and they are inherited by humans. One of these will be inherited by an individual. So these 9 will give 4 blood group type A, B, AB or O. So this is an example of multiple alleles and it also exhibit codominance. Now codominance it refers to heterozygous condition. For example, if you see these A, B, these are heterozygous. However, both are capital and the reason for that is both alleles are expressing in the heterozygous condition or in hybrid and their product or phenotypes can be measured or detected. This is known as codominance. So this is a very good excellent example of multiple alleles and codominance. Just to test your knowledge on multiple alleles, I have here two queries or two statements. You have to select true or false. The first statement is, in humans, each body cell has a unique form of an allele. The correct answer is, this is a false statement. Second statement is, in an individual, all body cells have the same allelic form of a gene. This is true statement. Thank you for watching this video. I request you to like it, share it and subscribe it. Until next time, you take care. Bye bye.